myself now?
Sure. So for some reason the mail bounces back, but I think it's okay. We'll figure it out.
Again?
Yeah, for your email also.
Maybe you can just present it from your side.
Yeah, well, I'll try, but no worries. Let's see what happens.
Yeah, my screen is visible. Yeah. Perfect. So we are live now, and thank you so much, Dr. Amey, for joining today's session at the SciComm Synapse. And we will try our best to convince our audience with your discoveries and also your journey. And also, I hope that it motivates a lot for all the new generation and upcoming scientists today. So, with this, I kindly request my teammate, Bhakti Sadhguru, to please introduce you today.
Thanks, Dhairya.
Yeah, very good morning to everyone. It's truly an honor to introduce today's speaker, Dr. Amey Redkar, a highly accomplished scientist and academician in the field of molecular plant microbe interactions. Dr. Redkar is currently working as a Reader F Assistant Professor at the National Center for Biological Sciences located in Bangalore, Karnataka. His academic journey is as impressive as his research accomplishments. He completed his PhD in biology from the Max Planck Institute for Terrestrial Microbiology, Germany, in 2015 as a DAAD long-term fellow. Before that, he earned his master's and bachelor's degrees in botany from Savitribai Phule, Pune University, laying a strong foundation for his future work in plant sciences. Dr. Redkar's research focuses on plant-pathogen interactions, particularly understanding how fungal pathogens like Fusarium, oxysporum invade and manipulate their plant hosts. His work explores molecular mechanisms of host manipulation, chromosomal plasticity, and root colonization strategies, contributing significantly to the field of plant pathology and host pathogen biology. His excellence in research has been recognized through numerous awards and fellowships, including support from EMBO, FEMS, SERP, and DST, among others. He is also committed to science outreach and mentorship, from supervising postdoctoral and master students to instructing international workshops and practical courses. He has played a significant role in training the next generation of scientists. He also regularly engages in public science communication, including initiatives like Skype a Scientist. Currently, his collaborative work spans across leading institutions in India, Europe, and United States, showcasing the global relevance and integration of his research. Beyond the lab, Dr. Redkar is known for being a thoughtful mentor and a collaborative scientist, and someone deeply passionate about bridging research and society. So, with that warm and detailed welcome, I now invite you all to hear directly from Dr. Amey Redkar as he takes us through the exciting world of plant pathogen interactions. Now, I'd like to hand over the session to Dhairya to continue the event. Thank you.
Thanks so much, Bhakti. Thanks so much. I think it was a super detailed introduction, and thank you very much for this opportunity to interact with all of you. It's a pleasure, and as I said, kudos to all of you in terms of sparing some time to make this professional community and linking all of us worldwide, which is very much appreciative in terms of exchanges and super important in science communication. So I very much thank you for this invitation and look forward to the interaction.
Thank you so much, Dr. Amey, for joining today's session and accepting our invite at the same time. So let's begin today's session. So the first question that I would like to ask you is that can you share how your international academic journey, spanning countries like Germany, UK, Spain, etc., India, has shaped your perspective on interdisciplinary research in plant biology?
Yeah, so I would start saying that I'm classically a botanist by training, and I did all my undergrads, as Bhakti mentioned, at SPPU, which is University of Pune.
Mm,
I did the specialization in cytogenetics and plant breeding, and I was super hooked by understanding plant pathogen interaction during my master's project. So that made me to further join one of the research institute in Pune to gain some experience, wherein I was fascinated by the system which I was investigating. So I did apply to the DAAD fellowship during that time. That's a German academic exchange service that allows you to write a full grant application for your PhD studies. So I ended up in the Max Planck Institute for Terrestrial Microbiology. So
mhm,
I had a 3 years fellowship with me, which was great, allowing me to start my PhD studies abroad. So
mhm,
I did my PhD in Max Planck Institute in Marburg, which is a small town, a wonderful town very near to Frankfurt.
yes.
For my postdoc, I decided to move to England. So
Mhm,
I did apply for this EMBO, the European Molecular Biology Organization. It's another wonderful organization that funds super competitive fellowships for Europe.
mm,
That gave me a chance to move to England, and I was in Norwich at the Sainsbury Lab, where I did my first postdoc, a little bit outside my comfort zone to shift
mhm,
again to identifying plant resistant genes. But I quickly realized, okay, that's not a subject which I could go on for a
mm,
long time in my career. So
mm,
I did write another Marie Curie fellowship, which is another program in Europe, which allowed me to move to Spain, again at the university setup in University of Cordoba. It's a wonderful city south of Spain, very similar to that of the Indian weather, which I was very happy about. But then the project was mainly shaped in terms of initiating a line,
mhm, mhm,
which I would build my whole long-term career on. So I started to work on the fungal model that we currently work on in Spain during my postdoctoral time.
mhm,
Did establish a line of research utilizing my postdoctoral first experience, and
mhm,
then I was in Spain for around 4 years time. After which I decided to move back to India, and then was very briefly on a Ramalinga Swami fellowship, where I returned back to India at Pune
mhm,
University, back to my home place. And then I joined NCBS from March 2023. So yeah, I think this journey across different countries have really given me a global view of science. Let's put it that way, and allowed me to broaden my horizons in terms of thinking from local to global scale, and how science works in terms of international teams, exchanges, and science is truly global, and it needs to be global to
yes.
achieve something highly impactful. So that's my snapshot of my journey across countries.
Fantastic. I would say it is an honor to talk to a scientist which is recipient of almost all the international European fellowships like EMBO and DAD and Marie Curie and all three, I would say. So it is an honor. So, like, since you mentioned about your journey spanning so many countries and working in different academic settings, like what motivated you to specialize in fungal pathogenesis at the first place, and particularly focusing on Fusarium, and what challenges have you faced in the niche
Yeah, so
here?
as I said, I think I would say that my initial master's project was a turning point, which
Mm-hmm,
really made me super interested in a project of plant microbe interactions. So this was a
mm-hmm,
very basic master's project. I think not even a full-term master's project. It was a few credits just as a part of my fourth semester. But I think the subject was too interesting, and I give the full credit to our
mm-hmm,
teachers who are super passionate about in terms of delivering that subject to us. So I think which
mm-hmm,
was certainly a point to generate curiosity, that this is a topic which can definitely interest me a lot. And I would say that, yeah, I mean, after I took that as a challenge, then my always career steps have been around plant and pathogen interaction, exploring very different systems in that go. Also, I think it was a topic which was gaining attention, or I think it is a topic which can be well connected to societal needs, because plant pathogens are on the rise. Changing climatic conditions makes it super important to understand how these diseases progress, how pathogens have been so successful, and so on. So I think the connectivity of the topic to general public, as well as I think the efforts taken by our teachers in the initial stage to really deliver that kind of interesting aspects to us, really change the mindset in terms of pursuing something for long term. Talking about coming to Fusarium, I think it was, again, as I said, my time in England was a little bit outside my comfort zone, wherein I decided to take some experience on Arabidopsis genetics. So Arabidopsis is a very model plant. It's like, let's say, like the HeLa cells of the animal world. And so I think it's a wonderful plant wherein genetics is easy as compared to other crop models. I wasn't having any experience in it, and that's the reason I thought like that would be a wonderful opportunity for me to gain some experience. But I soon realized that this, even though being a wonderful model, is not really my interest to pursue for very long term. And the reason to choose Fusarium is mainly because Fusarium does adapt to both the Arabidopsis. It can colonize Arabidopsis, as well as it's a very economically important pathogen. So it can drastically destroy all your crops. So this, I think, is like, again, interest for the system that caught me up in terms of saying, okay, now I can use the experience that I had during my first postdoc and do something wherein I can really understand some important questions which are of pressing need. So I think that was just a thought which fascinated me to understand, okay, this gives a chance to explore what I have learned previously. And hence I came to Fusarium.
Great, great. So, uh, so Fusarium, like on field, if I say, then how do your discoveries about the transcriptional plasticity in fungal pathogenesis influence the current strategies of the crop protection on field in agriculture or from day to day life? Let's
Right.
say.
So this aspect is like a very recent study that just came out as a preprint on from my lab here at NCBS, and I think we were behind this question for the last few years time now, and I think the main topic of investigation was this fungus is pretty notorious. So what it does is, let's say, it infects very specific plants by killing it. But in the absence of crop, it can survive almost in any plants that are present in the field. It means that you can naturally think of when a farmer grows any particular crop. Of course, that crop is not present in the field all 12 months, right? I mean, there are crop cycles wherein the different crops are sowed in the field across an year. But fungus like Fusarium has adapted to really colonize and infect plants. Any plant it could be in the field. It is an opportunistic pathogen. It means like it can survive almost in any plant without killing it. And as soon as it finds its host again, it kills it back. So this means this was the point of fascination that we thought, oh, why not? We should study about what makes the fungus so successful in terms of adaptability towards this diverse host range. And then we studied about that interaction, which also has a very different perspective about, okay, how did the fungus learn to adapt? How did it become pathogenic? Was it pathogenic right at the beginning, or it learned across its journey across different crops to be pathogenic at one point? And what are the kind of mechanisms that the plant has evolved in terms of really identifying and distinguishing, because several isolates of Fusarium are also helping the plant, right? They are called as endophytes, and they do not cause any pathogenicity on any plant. Rather, they help the plant to grow. and protect it from pathogenic strains. So this means that the fungus is both a helper and a destroyer. Yes, I think it's very interesting to understand this, let's say, trade-off as to what is the trade-off between a plant and a microbe that colonizes it so intensely.
I see. I see. I could understand, very nicely this concept of plant and parasite interaction. Rather, I would say host microbe interaction here, because, like, I used to, I used to visit my father. He also works in the agricultural and plant biology field in tissue culture lab. So I have been hearing this thing since couple of years now,
Right.
and then and great. So, like, since you mentioned about that, how this plant, plant host pathogen interaction switches from one, helping nature to its parasitic nature here. So, did you find in that paper as well? Is there any switch between this, and when exactly do they switch from helping nature to the parasitic nature?
Yeah, so this is a long-standing question in the field
Mm-hmm.
of plant microbe interactions. We, I think,
Mm-hmm.
have a very limited knowledge as of now as to
Mm-hmm.
what are those signals that really trigger the switches
Mm-hmm.
between a helper to a destroyer or the other
Mm-hmm.
way down, which
Mm-hmm.
is very less. But I think what we have found out in that paper is we at least found a specific gene locus and a cluster
Oh.
of genes that really helps the fungus to adapt to these diverse plant species.
Mm.
And diverse in
Mm.
the sense we are talking about the plant species that have diverged apart around 450 million years, right? So one is not having any vascular system at all.
Mm-hmm.
The other does have a vascular system. So I think
Mm-hmm.
we have shown now that this fungus has a set of genes which helps it
Mm.
adapt to very different plant species, which are
Mm.
in terms of
Mm.
an evolutionary perspective.
Mm. Mm.
On the other hand, I think root is pretty complex to investigate. One can think about it's like a gut, right? And it's like
Mm.
a microbiome. Everyone is present
Mm.
there. It's like a party site for everyone.
Mm.
Because the plant also has to choose its beneficial microbes.
Mm.
So dealing with Fusarium is not that easy for the plant
Mm.
because it's
Mm.
also causing health benefits for the plant if it
Mm.
activates its immunity constitutively and always.
Mm.
So because of which the plant has to be extra smart in terms of really activating its immunity to
Mm.
fungi such as Fusarium, which have adapted to colonize the very deeper layers like the vasculature.
Mm.
We have very limited perspectives about this as to what are those switches that determine pathogen to non-pathogenic or
Mm.
non-pathogenic to pathogenic switches
Mm.
for killing. And that's one of the
Mm.
things that we actively investigate in our lab here at NCBS. Yeah.
Mm. So, like, gut microbiome, root microbiome also does exist, and I am informed a little bit about it, not so much. But do these root microbiome, other parasites, or I would say a symbiotic relation bacteria or pathogens, also support the fungal species for
Yes.
the infection?
So yeah. So if the deeper you go into the topic, it's like
Mm-hmm.
exact trade off, and it seems to me like one can give an analogy of how human trade offs are in terms of business. It's
Yeah,
like business in terms of for
exactly.
resources, for
Mm-hmm.
nutrients, for
Mm-hmm.
space, for accommodation.
Mm-hmm.
Let's say for forming alliances together. So I
Mm-hmm.
think we do some work in the lab wherein we dissect the plant microbiome.
Mm-hmm.
So we have done like isolated compartment specific microbiome, which is culture
Mm-hmm.
dependent, meaning that you can isolate microbes from soil. Of course, you can culture only a handful fraction of microbes from whatever are present in the soil. But then
Mm-hmm.
we can go back to do fungal genetics. We
Mm-hmm.
can do
Mm-hmm.
reporter lines of Fusarium, which can
Mm-hmm.
express any reporter, for example, GFP or RFP and so
Mm-hmm.
on. And then by high throughput screening, we can understand which are friends of Fusarium, which are enemies of Fusarium,
Mm,
which are strangers, which are predators, and so on and so forth.
mm,
And then what
mm,
we can do is we can try to engineer microbiome based on making small communities, or let's say
mhm,
small synthetic formulations of different
mhm,
members of a community, and
mhm,
then understand how these different communities interact with each other in the presence
mm,
of fungus. So when there is a fungal attack, the plant is also
mm,
clever enough that the
mm,
signal is transferred to only a cluster of community, not everyone. And it makes sense because, let's say, for example, in the case of a problem, we have very designated roles, who is going to do what, right? And
yes.
I think in case of emergency, we act and we have standard SOPs.
Mm,
So it's very similar to that. In case of a fungal attack, I think the plant transfer its signal to very specific community which does the job of coming together and then
mm,
transferring the signal to others. So I think we have identified very specific bacterial communities which do a job only in maintaining plant fitness. There are sub-communities which only act in terms of defending the fungal attack. But there are also communities who have been cheaters and being on the fungal.
mm.
It's also very interesting to understand how these bacterial members from the
Mhm,
microbiome have gone on the fungal end, and how
mm,
fungi, what kind of benefit does the fungus give it to them, and,
mm,
and what kind of utilization of this long fungal-like channels
mm,
bacteria use to migrate on the plant surface. So yeah, it's like Monopoly, playing Monopoly. Let's say.
mm. This is quite fascinating topic of always it. I mean, it has been always a fascinating topic to know how the microbiome, let it be in human or plant, how these different bacteria they work together for even betterment and also for against the host system. So this is quite interesting. So, your work often involves advanced visualization techniques to understand various mechanisms of fungal pathogenesis. So, what technological breakthroughs have you been impactful during your studies of your entire, I would say, career, academic career, and also how did you see the growth of technology in your field, especially?
Yeah, so I think we cannot isolate a specific field as such. We all
Mm-hmm.
know that technology is advancing super rapidly. Especially, I can comment more in the context of biological research, let's say,
Mm-hmm.
rather than my field of
Mm-hmm.
plant microbe. And we
Mm-hmm.
all know that sequencing is growing very very rapidly. And I think
Absolutely.
we also are aware that there are huge data sets which people have
Yes.
been doing. Also, COVID has made us aware in terms of large scale population studies, also helps a lot in terms of understanding the evolution of pathogens
Hmm.
or microbes. So, in that sense, certainly the field that we are in also has seen lots of developments, particularly
Hmm.
in terms of structural biology. Let's say, for example, understanding the parallels between plant and animal immunity, and what are the commonalities between the two in terms of signaling cascades which activate immunity downstream. We know that there are many similarities across plants and animals as to how both of them use the NLR proteins, for example, in terms of activating immunity,
Hmm.
how they form those inflammasomes or signaling complexes that initiate cell death
Hmm.
mechanisms.
Hmm.
One of the growing aspect in our field is also understanding the interaction at cell-specific resolution. That is something which is gaining a lot of attention because I think, as
Hmm.
I said, root is a very complex tissue to work
Hmm.
with. It has 16 different cell types and all developmentally different programs, as well as defense programs are very different according to the cell types you are looking at. And hence I think understanding these kind of interactions at cell specific level or at cellular resolution is much more let's say realistic to understand
Hmm.
how the pathogens adapt at a micro scale, and then
Hmm.
the type we see at a macro scale. So this is something which is rapidly growing, and
Hmm.
this I feel like is super fascinating to understand. It's still emerging field of research wherein we are trying to understand how different pathogens adapt to different cell-specific layers, whether plant immunity also makes a
Mm-hmm.
decision at one specific point when plants
Mm-hmm.
are not able to tolerate that interaction. Of course, the
Mm-hmm.
interactions are very, let's say, intimate and sophisticated because
Mm-hmm.
these are all microbial cheaters which mimic
Mm-hmm.
plant as beneficial microbes to take resources. And hence, I think it's very interesting to understand what these trigger points are in terms of
Mm-hmm.
activating immunity, right?
Mm-hmm.
And that's growing on. But with the technologies, it has become super relevant that also people are versatile in terms of
Mm-hmm.
being open to understand computer languages, data analysis.
Mm-hmm.
It's important to also learn tools to analyze the data that we generate, and
Mm-hmm.
because data generation has become way easier than the analysis of the data, and hence
Yes.
I think makes things more challenging for us also to be open in terms of going outside our comfort zone to make relevance of
Mm-hmm.
that data into addressing our biological questions.
Mm, mm. Great. So I would quickly pitch in one small question. Do you find differences? So this is also for really for the young scientists and upcoming, future generations of our country, and also, I mean, people who want to move ahead in STEM and research. So what differences you find in, you know, pan countries, let it be India or European countries or maybe other countries, in terms of technology and science or research, I would say, or utilization of the technology?
Yeah, I think there are certainly differences. We are let's say in the line of development. We are a
Mm-hmm.
developing country, and
Mm-hmm.
some of the countries are already developed. So certainly, I think a one-to-one comparison is very difficult when you say, okay, certainly
Mm-hmm.
you will find some advantages in there, but I
Mm-hmm.
would say that we are actually developing in terms of being associated with several
Mm-hmm.
uh, global programs, especially in the
Mm-hmm.
context of EMBO, which I would say
Mm-hmm.
India is an EMBO member. So
Mm-hmm.
the audience, whoever doesn't know, you are eligible to apply for all programs of EMBO in terms of its fellowship, exchanges, and so on. We are a part
Mm-hmm.
of HFSP, the Human Frontier Science Program. So India is a part of that. So you are eligible to apply for all that programs. Commonwealth, we are eligible to apply for all Commonwealth programs. Certainly, there are differences in terms of let's say the kind of projects that are handled. But I would say we have our own unique niche, especially focusing on, for example, antimicrobial resistance or
Mm-hmm.
tropical plant diseases. This is something that
Mm-hmm.
are present as a niche and much more needed to be resolved. And these are national problems to be resolved in the Indian context. So
Absolutely.
they have their own advantages. What we can take from outside is more interdisciplinary nature of the projects. So I think we will definitely have to learn from the Western countries about going deeper into a problem that we have initiated to resolve, right? So it just cannot be superficial in terms of handling projects. It has to be until the depth of the problem, so that it can really serve the purpose with what it was initiated. So that's
Mm-hmm.
something to learn from the countries outside. But I think we will
Mm-hmm.
have our own niche and our own important problems that we have to address much more.
Absolutely, absolutely. I agree on this, Dr. Amey. So, what lessons from your Marie Curie fellowship experience would you pass on to the early career scientists considering joining similar programs?
Right. So I would say not very specific to Marie Curie, but let's
Mm-hmm.
say to any independent fellowship.
Yeah.
I have always been a strong advocate of writing your own grant, because
Mm-hmm.
I strongly feel there you are your own boss, right? I mean, it's
Mm-hmm.
your money, it's your award. So
Yeah.
I feel I strongly insist anyone in the audience who are thinking about writing grants to start with first travel grants. They are easier to apply for.
Mm-hmm.
So if you haven't done anything on that lines, please do. There are lots of travel grants for attending conferences, also for short stays with your collaborator,
mm,
and so
mm,
on. You can
mm,
start with that. EMBO, Marie Curie, or DAAD are competitive schemes of fellowships.
mm, mm,
Certainly worth trying. The success rate for any of these fellowships is roughly around 10 to 15%. So you can imagine the kind of competition which is present. But I think you need a very clear objective or a very clear focus as to why you are writing that application and what's your question that you would like to address. The
mm,
complementarity of your host also does matter in this kind of grant schemes, meaning that why you are the right person and why that host who is supporting for your application is the right host for you to go
mm,
to that lab, and do both of you complement each other in terms of solving that proposed problem.
mm.
Also, I think you can take ownership of your results as well as failures, and this
Yes.
gives a very good opportunity for you to learn how to progress on your independent path. So
Mm,
that's, I think, very helpful in terms of writing independent grant applications. They are rewarding, and they are equally rewarding, and it's always a high risk, high gain uh, kind of kind of alternative.
mm.
I very much insist each and everyone in the audience, whoever has a strong desire to apply for independent funding, you should go for it always, because I think that's
Mm,
how the CV will be built at one point. If you want to if you want to make a career in any
mm,
academia.
mm. I would say this is a very nice and very important tip, which I would also personally accept and imbibe it in myself to start writing a personal grant. Let it be a very small travel grant in some abroad country other than EU. So, as someone actively involved in public engagement and science communication, how do you bridge the gap between the complex plant microbe interaction research and the public understanding, or rather, I would say that on site to the farmers or in the agricultural field?
Yeah. So as I said, I mean, this topic is not that away from the general audience. So I
Hmm.
would say that
Hmm.
okay, the topic is familiar to general audience. People read a
Hmm.
lot about the crop decrease, emergence of a
Hmm.
particular disease, and so on. So rather it is easy to connect to general audience. The problem
Hmm.
is the misconceptions, because there in this kind of topics there are several misconceptions that people have in terms of, let's say, genetically modified organism,
Hmm.
a crop improvement program. So
Hmm.
I think there are several misconceptions among the general audience that need some kind of trust, and this is very challenging to explain. I think it has its own advantages and disadvantages. Also, in terms of, one of the important aspect is to connect yourself way early in schools. For example, we are doing a program wherein we are really understanding and delivering the basic concepts of how plants do have immunity and how
Yeah.
to fight against invading pathogen in a school, in several schools across Bangalore, by doing a comic book. This is in process and will soon be available online as well. So we have created a comic book to supported by India Bioscience,
Mm-hmm.
and we would like to really advance, or we would like to really promote the basics at the level of high
Hmm.
school. Uh,
Hmm.
to reach to farmers is another new challenge. So I think that
Hmm.
is, there are several traditional practices which farmers have already implemented, right?
Hmm.
And then you
Hmm.
need, it's more structured in terms of, if you want to implement something with farmers, you first need to gain the trust that they have,
Hmm.
and you
Hmm.
need to justify them why they should try that in alternative to what they are already trying. So it's
Hmm.
very challenging, but I think the farmers, there are always some people who are very enthusiastic to try new tools and concepts, which particularly researchers tell them. And once I think it is spreaded in the farmer community, it makes things way easier to try something at translational level. Uh,
Hmm.
So I think it's, yes, outreach is fun to deal with. It's also super rewarding and
Mm.
incredibly rewarding in terms of being really translating your findings to someone or knowledge to someone,
Yeah. Mm.
factual knowledge to someone. But at the
Absolutely.
same time, it's challenging and worthy to gain trust, and entirely depends on trust.
Absolutely. I think this is very important to connect your science, what we are doing in the lab. Maybe the future generations would also know, and also translationally speaking, the application is very important. So, with a significant number of publications as corresponding author, how do you balance research responsibilities and as well personal responsibilities, and also together the mentorship and the collaborative endeavors?
Yeah, well, I think it's a very difficult question to comment. I think the point I still feel it's always challenging with limited time. If you ask my
Hmm.
lab people, they might be say I am busy all the time. If you ask
Hmm.
my family, they might say the same thing.
Busy all the
So
time.
I think time is limited. Certainly, we try our best to give time to both. In terms of publication, certainly that's the kind of output that is needed in academia.
Hmm.
Unless you
Hmm.
publish the work that you do, it's pointless because the community needs
Hmm.
to know what you do. In terms of also peer review your findings. So I think certainly publications are important, but I rather feel that mentoring is much more important, especially for a lab like us, which is a new lab.
Hmm.
Again, that to really create uh, good scientists and good people who can take the science ahead. So mentoring is much more important to me than publications at this level. Uh,
Hmm.
and certainly I feel like every individual that comes or runs through my lab is very different in terms of their nature, their qualities, the things they bring to the lab. So I think it's equally important to understand their career stages and their
Hmm.
interests and their next steps or desires, what they wish for. So I think we try to work around what their plans are and where what their aims are.
Hmm.
Of course, a lab journey is a very short time that they want to progress in their careers, and I think it's really fun to mentor these young people coming through the lab, and then to really deliver impactful science, which only happens when every individual of the lab is happy.
I see. Really, I mean, it's not just the from you what I hear today. It's not the job of the mentee to really just join the lab and work, but also the mentor to how it will shape the mentee's future now in the lab or maybe outside the lab. You know. So, moving on ahead, your recent keynote at Fusarium community meeting emphasized on transcriptional plasticity. So, what key message did you aim to convey to the community?
Yeah, so the point is this is very specific to Fusarium community. So this
Mm-hmm,
is a
mm-hmm,
community that meets once in 2 years. Last year it
mm,
was in
mm,
Silomar in near to San Francisco, and
mm,
I think the entire community is again addressing Fusarium pathogenesis, or particularly let's say vascular wilt, which are well developed pathogens. So what we are trying to understand is how do these communicate? How do these pathogens survive for a very long time? So you
mm,
would be amazed to hear that the spores they make can survive up
mm-hmm.
to 20 years in soil. So without any
20.
problem, right? So you can imagine how deadly this disease might be. Once present in the soil, it's completely impossible to eradicate it
Mm,
because it's
mm,
tricky. And hence, I think the way forward is to understand again how these microbes communicate, what kind of trade offs they have, what kind of different microbiomes exist across different sites in the world. And depending upon okay what are the adaptation strategies of these pathogens in terms of different crops and the new emerging properties,
uh-huh,
that is something that we are working hard now. Of course,
Mm-hmm.
as a community in Fusarium, and also I think gaining help from technology, we that things make our work much more rapid. So we are understanding what happens at an entire genome level rather than focusing on individual genes. We
Mm-hmm.
talk about entire genome level and how things are changing at the level of pathogen or at the level of pathogenic system rather than targeting. Of course, individual genes have their own importance,
Mm-hmm.
but gaining a snapshot of the whole pathogenic genome makes things much more broader and easier to understand.
Mm-hmm. I see. So, what trends in the fungal genetics or plant pathogen interactions do you predict would be on demand, I would rather say, in the next upcoming decade?
Yeah, so it's always a challenge to work on plant pathogen interactions because these are
Mm,
co-evolving systems, meaning
mm,
that
mm,
you are doing research on the unexpected, right? And you don't know how these both system co-evolve. So whatever
mm,
we are working on the plant side, and of course, as a translational perspective, there is much interest to work on the host rather than
mm,
on the pathogen. But pathogens give us many more surprises because the genomes are super compact and they are evolving at a very different pace as compared to that of the plant, right?
mm,
So I think the challenge is to really keep ourselves at the pace of the microbes. And the challenge is again gaining many novel insights into understanding how pathogens infect plants, whether there are any key nodes in the plant signaling network that the pathogens
mm,
target as pressure points, let's say,
mm,
which the pathogen is really needing in a crucial means to establish itself in the plant.
mm,
This is
mm,
something that is gaining a lot of attention at a system biology level.
mhm,
And certainly, microbiome is also one of the important aspects that the community is trying to investigate, particularly in relation to agribiological and bioformulation, sustainability. These are
mm,
all very emerging hot topics, and I think we cannot ignore them.
mm. I see. So, how has your participation in platforms like Skype a Scientist or Norwich Science Festival impacted your approach to communicating science to general audience or students especially?
Yeah, very much, very much. I think that has given really an opportunity to
Mm-hmm.
introspect myself, because
Mm-hmm.
I think Skype a Scientist was a wonderful platform wherein I have done this to explain my science to school kids of standard fifth or standard lower than that, fourth, right? And it's super challenging to convey our science on what we do uh, for these kind of small audience, right?
Absolutely.
It's very difficult. So I think that has given a very nice opportunity to introspect the kind of
Mm-hmm.
language we use. It's very much helpful to use always an analogy. No one cares
Mm-hmm.
about your scientific words when you
Mm.
want to convey your science to a general audience. It's super
Yes.
important that you reach their level and what they know,
Mm-hmm.
right?
Mm-hmm.
So
Mm-hmm.
if you start a sentence by, if you can imagine a forgotten strawberry in your refrigerator, uh,
Mm.
one has seen a infected strawberry, right? When you forgot it in
Yeah.
the refrigerator,
Yeah.
it's
Yeah.
always covered with a fungus. So it makes
Mm.
things much more connected to you when they are explained to you in your everyday living, rather
Mm.
than talking about genes and what pathogens do and how they infect. So Skype a Scientist has been a wonderful program. It was where I participated, and it has allowed me to really brush my communication skills in terms of
Mm.
going to the level of the audience that and connecting them to their everyday living.
Mm. 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 Great. So, can you discuss how funding and fellowship opportunities such as Ramalingan Swamy Re-Entry Fellowship have influenced your ability to establish a robust research program in India?
Yeah, certainly. I think Ramalinga Swami or Ramanujan, for example,
mm,
by NRF now are very wonderful programs that provide
mm,
opportunities as springboards for the
mm,
early career researchers to come
mm,
back to India. They provide
mm,
a platform for you to at least start your research or kickstart your research. So I think in that sense, certainly they provide a channel to return back to India.
mm,
Uh,
mm,
well, thinking about a long-term program, I certainly feel everyone applying for it should know that they are fellow positions, right?
mm,
Meaning
mm,
that you also need to be aware of how to transition from a fellow to a full faculty positions. And I
mm,
think
mm,
that also needs to be aware what your long-term programs are, what your plans are in terms of asking or discussing with your host who have provided you
mm,
host for a fellow positions. What are the opportunity exist in that host institution to establish your long-term research program?
mm,
I think there
mm,
has been some revisions in terms of Ramalinga Swami, in terms of its in terms of its timeline. I think initially it was for 5 years. Now they have made it for 3 years time, if I'm not wrong. So I think that gives you, like, let's say it's an independent position, which can
mm,
be said to be like a little bit higher from a postdoc, where you're completely
mm,
independent and trying to establish your own line of work, right? By also hiring people. But at
mm,
the same time, you should be aware of the transitions and the options that are present to establish your long-term programs, which is also crucially important. But no doubt,
mm,
these two programs have been a flagship of our country and have certainly attracted many talented researchers to come back to India to
mm,
implement their research goals.
mm, mm, mm. Great. So, since you mentioned about transiting from a fellowship to a faculty level position, how does your current work at NCBS integrate the global research collaborations, and also what goals do you have in expanding your network across India and as well as globe, and also how do you keep your host institute in center of all of this?
Yeah, so as I started my talk, that or whatever, the first answering the first question, as science is
Mm,
global, and I truly
mm,
believe this, that science is global, and I think
mm.
we to solve some of the question, we need to have some of the people who are experts in it. I
Mm,
think my current institution, National Center for Biological Science, provides a wonderful opportunity because
mm,
we are different biologists working in very different streams of work.
mm,
I think we are establishing programs with, for example, physicists to understand what physical aspects are involved in plant microbe
mm,
interactions, either it be
mm,
during host invasion or what does a microbe decide in terms of expanding its developmental aspects, what kind of physics is
mm,
involved in there. We also have at our institution the Physics of Life as a very flagship program, which is very running. In terms of
mm,
my global connections, certainly they are very important, and we are pursuing several projects in collaboration with different European institutions. One
mhm,
of the recent program that we have initiated just a month ago is the Max Planck Partner Group. So we are a Max Planck Partner Group partnering together with the Max Planck Institute for Molecular Plant Physiology.
mhm,
And that program allows us to team up with another team there who is interested mainly in understanding beneficial microbes. And
mm,
we together are mainly understanding what are the overlaps and what are the similarities, whether pathogens have hijacked some of the symbiotic pathway. So everyone is familiar with mycorrhizal fungi. So generally
mhm,
people know, yeah, mycorrhiza are good fungi. So we want to understand whether pathogens like Fusarium have adapted some strategies to hijack the symbiosis pathway or
mm,
nitrogen fixation, for example, and whether they have hijacked that engines to be so successful
mm,
at pathogens. So this is a program that has just started in my group, uh,
mm,
and a partnership program, and hence such kind of things allow opportunities to make your science much more global, right? And also with the experts on board who have a knowledge on that.
mm. It's very interesting to listen to you and talk to you about a lot of many things. But due to time constraints, we have to, make it in parts, definitely. So my last question for today from my side would be, so I asked this question to almost all, I would say mostly everyone who, so all the guests who join our events as guest speaker. And the question is that what is your favorite discovery in science until now? I understand that it would be very difficult to choose one, but maybe here I would push you to choose one discovery that so far has interested you a lot and seem that it's a breakthrough in the science.
Yeah, I think as you rightly pointed out, I mean it's very difficult to choose one. And
Hmm.
that too, I think you make the question much more tougher, saying that in science rather than in whatever your field or technology. So I think it's completely difficult to say that okay, this is my favorite discovery. Certainly, one is always biased to his or her topic of interest.
Yes, yes.
And I think in my topic of interest, as I also describe, I think the structural biology or cryo EM is gaining a lot
Hmm.
of attention in terms of
Hmm.
understanding the interaction at a very molecular level of how this signaling cascade reacts. And that has opened new doors in terms of understanding translational perspective to really develop
Hmm.
crops, resistant gene cassettes, for example, clubbing together many resistant genes so that the crop is much more healthier for a longer span of time by different combination of resistant genes rather than only one.
Hmm.
So these discoveries have really opened across last 3 years have opened really new doors for understanding plant microbe interactions a lot. Yes. So I certainly choose related to my field, but there have been many other things which have progressed science, and I think it's hard to keep a pace with the upcoming papers every single day, right?
Definitely.
You as PhD students would know this way better than me as to how important it is, and it's impossible to keep with upcoming literature.
Definitely, definitely. So talking about cryo-EM, I have been quite honored, I would say, and I've met Professor Joshem Frank when he was here at University of Freiburg, and I loved the way he talks. And like as you correctly pointed out about the science in the very beginning, about the interdisciplinary aspect, about involving physics, biology, and everything, the cryo-EM discovery, I think, definitely suits
Right,
that.
right.
Great. So I think you can present your presentation now. We would, we would, we would.
So Dhairya, I
Uh,
just have sent you the slides. If you can just put
ah,
it up on your Gmail.
great, great, great. I would just have a look once. Sure. Mm. Yes. So it is on the community email ID. So I think one of the teammate will have to present it. Just a second. Uh, Dr. Redkar,
Yes.
can you please send the PPT to me once, like just responding on my email back?
Yeah, I did have sent to you.
Uh, okay.
So I
I
have
cannot.
sent to you. It's dhairya.sicom@gmail.com.
Yes. Mm, Unfortunately, I have not received it, but I can check in the spam folder. Yes, and here it is. Sorry. Mm, mm, okay. Perfect. And now I would present it. Here it is. Okay. Oops. There is something. Okay. I can go. And now I will present. Uh, I think now should be visible. Is it on slideshow mode?
Not yet.
Not yet. Okay. And now?
So I cannot see it yet on slideshow
Uh-huh. Okay.
mode, but
Yeah, okay. That is best. Yeah, sure. So I will just mute myself. Yeah. Okay. I will restart it. Okay. And now it should work. Yeah?
it looks good. Looks good. Thanks.
Perfect. Let's
So
go.
I think I'll be very quick. As said, I'll give a snapshot of my lab in 5 minutes. So
Mm-hmm.
our main objective is getting to the root of regulatory mechanisms in plant and fungal interaction. As you see, we are interested mainly in terms of understanding immunity in the context of that cartoon which is shown, which is a root transfer section. Our top biological questions are mainly understanding how pathogens evolve, novel virulent strategies
Mm.
to colonize complex tissues, how plants engage with beneficial microbes while at the same time they restrict pathogens. And we also are
Mm.
interested in understanding the microbe-microbe interactions and how they affect plant microbe in binary context, right? So that's the three main questions that our group investigates. Yeah, next. So, of course, microbial or any biotic interactions are very challenging to investigate. They happen above ground as well as below ground. Some are beneficial, some are non beneficial or pathogenic, and everything starts in the root. So, next click. Whereas you can see that this is where our food begins, right? And everything happens, much more interactions happen in root. It's a very complex slide wherein more microbes are present. You can imagine the microbes are super complex to understand in the root, and this is something which is hidden beneath. We cannot see what's happening underground. So in terms of the next slide, what you see is how biomass and activity of this soil microbiome influences the microbial interaction. The fungus that we work with, Fusarium, is certainly a soil-borne pathogen. So it infects the plant through soil and through root. And it has many things to interact with in terms of the microbial activities that are present surrounding the root. So what you see here is how intense are those microbial activities nearer to the root, and how they decrease as you go away from the root in their dynamics. So next slide. In terms of the system, we are using Fusarium oxysporum, which is a fungal pathogen, and that causes typical wilting disease. So this is what you see as a cartoon when you infect a tomato seedling. The plant is completely dead by the fungus when infected. And the colonization of the fungus, or the fungus senses mainly roots in the complex soil environment. The fungus can either be wilting or killing the plant, which happens very specifically when it lands on its host. But as I said in also one of my questions, the fungus can survive in any other plant without killing it, just as a right? And all this happens in the context of a complex microbiome. So just the next click, wherein we see that this is so complex. So many things are interacting together. So our lab is interested in understanding what is this trade-off and what dialogue exists to make the fungus so successful to enter this inner layers of the plant vasculature, right? Okay. So the program that we have in the lab is mainly focusing around three different themes. The first theme is about exactly understanding the crosstalk. What kind of dialogue happens between a plant and a microbe, right? What kind of things that the Fusarium tries to convince a plant to enter inside, and what kind of defense mechanism the plant makes in to block the fungus. The second theme of our program is mainly about understanding how the fungus senses where it has to go, right? And this program is mainly about, this theme is mainly to understand what are the kind of signals that it gets from the plant to colonize vasculature, and how does it know that it has to go inside a plant to colonize it, ultimately to lead to killing. And the third aspect of our program is understanding these dynamics. And we are interested in understanding how these microbe interactions are helping the fungus or blocking the fungus from establishing a binary interaction with its host inside the plant body. So these are the three main aspects of our program, which mainly fits into the molecular mechanisms which are underpinning microbial crosstalk and the host resistance and biological control, and how we can explore these aspects to ultimately reach plant resilience or climate smart crops. One of the other aspect is also to understand how this all got started and how this business of pathogenicity got evolved. So for this, we are establishing and exploiting Fusarium's broad host range to understand how did it adapt to vasculature in the first place. And we do this by establishing different models of plants across different lineages of plant evolution. So these two plants, what you see on the left and right, on the left is Markensia polymorpha, which is a plant which is non-vascular. It doesn't have a vasculature. It belongs to a lineage called liverworts, and which has diverged from our angiosperms, which are flowering plants, or like, for example, tomato. And it has diverged around 450 million years apart, right? And we understand this by establishing fungal pathosystem across these two divergent model, and then to understand what are the kind of genes that the fungus likes to utilize to establish associations with these two diverging plant, and what are the specific mechanisms that the fungus utilizes to kill a plant host. And as a part of this program, the next slide, in which we are basically understanding the evolutionary plant microbe interaction to understand the scenario of adaptation of fungus like Fusarium, whether the vascular colonization was an accidental pathogenicity. Did the fungus end up in that site accidentally? Did it happen as a relation of microbial competition with other microbes? And is it a quest for sporulation? We do not see any spore formation in the fungus unless it's colonized vasculature. So we do think that the fungus has likely gained this niche colonization for sporulating. And these are the aspects that are still under investigation, but we are interested in following up on the evolutionary questions. So a last slide in terms of the whole snapshot of our program is
Mm.
basically to understand how fungi adapted to first colonize the plant in the first instance. How did they make plant associations? What kind of
Mm.
genomic rearrangements made specialized lifestyle in the fungi to turn from non-pathogens to pathogens, colonizing inner layers like the vasculature? How this specific niche adaptation is shaped by the surrounding microbiome, and how these microbes help the plant to colonize very specific niches across flowering plants or angiosperms. So I
Mm.
think that's the end of my presentation, or a short snapshot of what my lab does. The next slide is just about acknowledgement, and this is my wonderful team that does all the work in terms of understanding the different perspectives that I talked about, and collaborators and all the funding agencies. And of course, I'm thankful to again for you to provide me this platform to present our work as well as my thoughts on my journey so far, and the audience who joined today. And happy to take questions if time exists or if there are any opportunities.
Sure, sure, absolutely. I think I loved the plant presentation, and also I feel that you're making the field kind of grow a lot and also attract more and more students into the plant biology. Definitely, no, Dr. Amay, I would greatly appreciate this uh,
Thank you.
initiative here. So, moving on to few questions, I have to switch between few tabs. Just a second. Okay, perfect. So, so we have few questions here, and Neha is asking that first of all, uh, she heard about the plant immunity. She never knew about it. She heard in a lecture, and she was really shocked to know that even plants have immunity to fight against the infections. So she's asking that what are the initiatives being implemented at or by NCBS for the undergrad students to for the exposure of the research environment in the field.
Right. So certainly, I think NCBS has several programs now in terms of getting all the not only plant microbe but every single topic that we investigate. We have something called as an undergrad lecture series. I think that
Mm.
runs around 2 months span on Saturday and Sunday, if I'm not wrong. So that
Mm.
wherein the PhD students and postdocs at NCBS participate in giving these lecture series. We
Mhm.
also
Mhm.
have our how to say summer school or even winter schools in covering some
Mm.
of the topics. Let's say, for example, physics of life or quantitative biology had a winter school. The same applies for summer school as well. There are also courses particularly related to microscopy. The Bangalore microscopy course is very famous for it in terms of learning and focusing on very specific tools. We
Mhm.
have also workshops related to proteomics and genomics, wherein anyone can apply for it and
Mhm.
participate. Other than that, I think as a part of our outreach programs, our communication office coordinates every month at least two visits. Generally, the
Mhm.
slots are very full, but I
Mhm.
think if your school or college is interested in visiting NCBS, and this is applicable nationwide, it's not limited to Bangalore. We have had visits from also Northeast institutions. So
Mhm.
we host two institutions to visit our labs as well as campus and give
Mhm.
an overview in what kind of topics that they are interested in. We tailor the visit
Mhm.
accordingly. Yeah.
Mhm. Mhm. Great. So there is one more question. So a lot of microbes present in the soil, in I would say in the natural environment, are unable to grow under laboratory conditions. How do you approach this problem considering fusarium per se?
Right. So this is a very valid and a very good question. As I said already in my microbiome aspect, we
Mm-hmm.
only can culture around 5 to 10%. 10 is also a lot,
Yeah.
mainly
Mm,
5% of the microbes. But
mm,
certainly, we also take independent approaches of doing independent sequencing where we know the complete microbiome, which are not culturable. So the point then is to see how we can engineer anything related to modulation or engineering does require a culturable microbiome. And hence, I think the aspect is to engineer microbiome which can rather act as a probiotic. Let's say giving a COVID vaccine, which would
mm,
basically trigger your immunity. So it's
mm,
something about playing along with the culturable microbiome rather than restructuring the complete unculturable, which is impossible.
mm,
So
mm,
that's what we do. We culture the
mm,
microbiome for synthesizing or recreating synthetic mini subcommunities, but that this is limited to only ones that are culturable.
mm, mm. I see. So we don't have much time to delve into so many questions, but maybe I would choose one more question, very important for students, I would say, and your advice would be quite useful to everyone. So, how can one know about the topic that is they are interested in and remain onto it without being distracted?
Oh,
I think.
well, that's a very let's say good question, which I don't know how to answer, but let me try to. So I think the point is there are two aspects of it, and
Mhm,
I would say that the first measure I would take is you should really introspect if you love your work, right?
mm,
Loving your work, or if you really feel happy about it, doing it, that likely points out that you are interested in a topic. But
mm,
I would say that this would eventually come. It's very difficult to say that you automatically know in the first undertaking that you are interested in an XYZ topic.
mm,
This will
mm,
eventually create your interest in a particular topic. If you like it, you will pursue it again and again.
mm, mm,
But there's nothing like if you talk about my journey, it has always been around plant microbe interaction, because I
mm.
realized at one point that unknowingly I started to take in projects which are related to the same theme.
Good
But it could
thing.
also very well be that there are people who have handled many different diverse topics and have
Mm,
ended up in a one topic of interest. So that is another route wherein you can try multiple things if you are
mm,
uncertain about, provided you are very confident about switching outside your comfort zones. So if that's the case, and if you are confident about yourself, there's no harm in trying multiple things unless you reach a decision that you are convinced about that that's a topic for your long term.
completely. I completely agree on this. I think this was a very nice piece of advice for everyone that you have shared today. And also, I would say uh, that today's session was really enjoyable and not many people are interested in botany. I would say I should not generalize this, but definitely there are less students going towards the field. But I think scientists like you are making the field seamless and also quite relatable for students to join in.
Yeah. Thank you so much. Thank
So
you so much, David.
thank you so much for accepting our invite, and we would surely connect more. And I would love to host one more session with you where we would delve more about immunity and plant microbe interaction, maybe soon. So I would kindly request my teammate to please proceed ahead with kind vote of thanks.
It is my privilege to extend a heartfelt vote of thanks on behalf of all of us here. First and foremost, I would like to express my sincere gratitude to our esteemed speaker, Dr. Amey Redkar, for sharing their valuable insights and expertise with us. Your engaging presentation has been both enlightening and thought provoking. It has left us with valuable takeaways and a fresh perspective. I would also like to thank all my teammates for their efforts in putting together this event so seamlessly. From coordinating the schedule to ensuring a smooth flow, your dedication behind the scenes has made this webinar a success. A special thanks goes to our audience. Your active participation, insightful questions, and enthusiasm have truly enriched this session. It is your engagement that drives the success of events like this. Once again, thank you all for being a part of this wonderful session. Let's continue to think, grow, and share together. Have a great day ahead. Thank you.
Thank you so much. Just for 30 seconds, thanks so much, Bhakti and Dhairya. It's wonderful to come on this platform again. Kudos to all your team for their enthusiasm to keep this running. This has been a very wonderful initiative, and I know that before me there have been fabulous speakers on this platform also to share their insights. And I think this is the only way to grow our professionalism and as well as professional network. So I think kudos to
Definitely.
all of you and good luck with all your initiatives. And I
Thank
look
you so
forward to
much.
interacting with you further.
Thank you so much, Dr. Amay. Thank you so much for joining today's session and gracing the platform.
Thank you. Have a good day.
Bye bye.
Bye.